All right, so here we're going to talk about the electron transport chain. And so the electron transport chain is made up of several complexes, and we'll go through each of these complexes. And so complex 1 is really going to take all the NADH that you generated in the TCA cycle and transfer those electrons into the electron transport chain. And complex 1 is referred to as Nadh dehydrogenase. It has within it a flavoprotein, the FMN flavoprotein, and the NAD, the electrons are going to be transferred to those flavoproteins. After it gets transferred to the flavoproteins, it's going to get passed along to your iron sulfur centers. So in your complex 1, you have 6 iron sulfur centers. So we'll have 6 of these. And so the electrons get passed from each sulfur centers. And then this complex is going to transfer to ubiquinone the hydrogen ion from NADH and a proton from the matrix. And this in turn, this complex will transfer 4 protons into the inner membrane space. And you're going to pass these on to ubiquinone. Now, there are some drugs that can interfere with this first complex. We have our barbiturate drugs, which are used for stress and anxiety. And the mechanism of action of barbiturate drugs are to inhibit the flow from those electrons from those iron sulfur centers to ubiquinone. There are other toxins also that can prevent this first complex. We have Rotonome, which is a pesticide, and this is going to actually complex with the dehydrogenase itself and inhibit oxidation of NADH to NAD. And so this pesticide will not only affect the electron transport chain, but it also affect those that have the dehydrogenase in the TCA cycle, such as the alpha-ketoglutarate, the pyruvate dehydrogenase. Those will also be affected. Now, an antibiotic that was isolated from a bacteria, pericidin, that is going to resemble ubiquinone. And so this is going to complex or compete with ubiquinone in picking up the electrons. And so all of these are going to have the ability to block oxidative phosphorylation at that first step. So it's going to block any of the NADH you made in the TCA cycle to enter the electron transport chain. Now, complex 2 is going to be part of the TCA cycle, so it links the TCA cycle and the electron transport chain together. So complex 2 is going to be the succinate dehydrogenase. It's going to be made up of 4 different protein subunits. The succinate dehydrogenase is the only membrane-bound enzyme of the TCA cycle. It has 5 prosthetic groups. It's going to have 4 different protein subunits, of which shown here we have C and D. We have here C and we have D. Are integral membrane proteins, and they contain heme groups as well as a heme B. So this heme B here. And also a binding site for ubiquinone. So ubiquinone will come in and bind to complex 2 as well. Now, one of the things to note here is the movement of the electrons. So the electrons are going to go from succinate, which is in the part of the TCA cycle. So succinate has its own binding site. Succinate will pass its electrons to FAD. So this is different than complex 1. Complex 1 had an FMN. And then FAD will transfer these through iron sulfur centers. So that's similar to complex 1. And then on to ubiquinone. One thing to note is that the heme, shown here, heme B, does not partake in any of the transfer of the electrons. However, if there is a defect of this heme in this enzyme, then you have an increase of reactive oxygen species. So it's believed that heme in complex 2 prevents the leakage of the electrons out before it can be transferred to ubiquinone. So oxygen is going to be the final electron acceptor of the electron transport chain. However, oxygen can flow freely across the membranes. So if oxygen is the preferred acceptor of electrons, then it's important to make sure that the electrons do not leak out of the electron transport chain because that would enable oxygen to grab those and make reactive oxygen species. Reactive oxygen species examples are going to be hydrogen peroxide as well as a very toxic superoxide radical. So again, point mutations in complex 2 near that heme B or the ubiquinone binding site are going to lead to some diseases associated with superoxide radicals, and it can have benign tumors in the head and neck of hereditary paraganglioma. Now, it's important to stop here because, as I said before, you have complex 1, which you can give the electrons from the TCA cycle NADH. We'll transfer them to FMN. Complex 2 is part of the TCA cycle. Well, succinate is going to transfer to FAD. And then each of these are going to pass them on to ubiquinone. Okay, so complex 1 and complex 2 will both pass them on to ubiquinone. Now, there are other electrons that need to be donated to the electron transport chain from different pathways. So one such pathway is fatty acid oxidation. And so in this case, you're going to have an acetyl-CoA dehydrogenase that will take the electrons and pass them to FAD. And then this is going to eventually also be passed on to ubiquinone. We also have the glycerol 3-phosphate shuttle from glycolysis. And so this is going to get passed to glycerol 3-phosphate dehydrogenase. And this is going to also be on the inner membrane of the mitochondria. And then we'll pass the electrons from glycolysis to the dehydrogenase. And then this will again go on to be passed on to ubiquinone. So ubiquinone is really a pool, and you can think of it as taking electrons from many different sources. So the pool of reduced ubiquinone comes from, again, that NADH, which came from primarily your TCA cycle. And then you had your succinate, and again, that's from your TCA cycle, and that goes into complex 2. You also have fatty acyl-CoA, so this is going to be oxidation of fatty acid. And you also have your glycerol 3-phosphate, which is through glycolysis. And then these are all going to donate electrons to the electron transport chain and reduce ubiquinone. Now the ubiquinone is going to pass its electrons to complex 3. And so your complex 3 is also called the cytochrome BC complex or ubiquinone cytochrome C oxoreductase. It's going to transfer electrons from ubiquinone, QH2, to the cytochrome C as it's transferring protons from the matrix of the mitochondria to the inner membrane space. Now the complex is going to be a dimer. It has monomers that create what's called a cavern. And within this cavern, you're going to be able to get movement of ubiquinone. So you can see here that we have our ubiquinone that's going to move through the cavern, and eventually it's going to donate its electrons to cytochrome C. Now, ubiquinone is going to go from the matrix side, QN, to the inner membrane space through that QP. So it's going to go like this, and then eventually you'll get it transferred to our cytochrome C. Now, cytochrome C is a soluble protein of the inner membrane space. It has a single heme, and it's going to accept electrons from ubiquinone in complex 3 in that cavern. Cytochrome C's purpose is to take those electrons from complex 3 and donate them to complex 4, and this is where you're going to get the final electron acceptor, oxygen. So in complex 4, you have cytochrome C to oxygen, and this is cytochrome oxidase. So the electrons from cytochrome C are going to be transferred to this enzyme and eventually find their way to oxygen to generate water. So this is the safest way for oxygen to be the final electron acceptor and not create radical oxygen species. And so it's very important that oxygen is delivered these electrons within complex 4. Now complex 4 is very large. It has 13 subunits. And it also is a proton pump, so it's going to pump protons into the inner membrane space as well. So within this complex, we're going to have 2 copper containing ions, and these are going to be complex with our sulfur groups of cysteine residues. And so here we're going to have our copper QUA, and here we're going to have another copper iron center, and that's going to be complex with another heme group. Now these copper and sulfur groups with that the sulfur comes from cysteine are going to resemble 2 iron sulfur centers of a typical iron sulfur protein. We're also going to have 2 hemes. We have group A and group A3, and we also have another copper, which is a copper B, shown here. And so all of these are going to be important for moving the electrons around and making sure it gets to oxygen appropriately. Now the flow of electrons are going to be through different subunits. We have subunit 1, 2, and 3. We're going to have cytochrome C, which carries an electron, 1 electron each. So we're going to need to have 2 reduced cytochrome Cs to donate the electrons to copper A. And then you're going to get another 2 cytochrome Cs to donate to another copper A as well. And then from copper A, they're going to go to heme A. And then eventually you're going to go to the iron copper center, the heme A3, and copper U B, or copper CUB. The oxygen will then bind to heme A3 and reduced by 2 electrons. Now you need a total of 4 electrons in order to generate water. And so you're going to have 2 more electrons come from cytochrome, 2 more cytochrome Cs that will follow the same pathway. And this is going to consume 4 protons right here. We're going to consume 4 protons from the matrix. And at the same time, we're going to pump out 4 protons from the matrix. Now, we also have the electron, the energy transport, and these electrons are going to be conserved, or this energy in the transfer is conserved in the proton gradient. So we have a matrix side of the inner membrane space and that cytosolic side. And so the proton gradient, because of the pumping of those protons by the electron transport chain, is going to be greater on that cytosolic side and lower on the matrix side, and so this potential is going to be used to generate ATP. So the proton motive force is this gradient due to the difference in the hydrogen concentrations. The electron potential that results from the separation of charges is when the proton moves across the membrane without a counter ion. So this is going to drive the production of ATP. Now, just as a side note, there are some inhibitors of the electron transport chain. So we have different sites of the electron transport chain that are going to be affected. We already talked about some that inhibited complex 1 to coenzyme Q. We also have a few that are antimycin C that's going to prevent the transfer of electrons from complex 3 to cytochrome C. We have carbon monoxide, which is going to prevent the transfer of electrons from complex 4 to oxygen. We have cyanide that's going to prevent the electrons moving through complex 4 to oxygen. We have dinitrophenol, which is an uncoupler that we'll talk about. Valinomycin, which is also a type of uncoupler. However, it is working through another charged ion, potassium, rather than hydrogen, but it's going to have the same effect. It's going to move the positive, and it's going to decouple the electron transport chain. And then there's a few others. We have atrocycloside, which is going to inhibit the transport of your ATP and ADP, so it inhibits that translocase on the inner membrane. And then we have oligomycin, which is going to inhibit the flow of the protons through the actual ATPase synthase.